Hello everyone, today we'll be discussing about the lactic acid fermentation. In lactic acid fermentation, the first step is similar to both aerobic respiration as well as ethyl alcohol fermentation. If you haven't watched ethyl alcohol fermentation, it's in our channel, go now and watch it. Right, so now let's talk about lactic acid fermentation. The first step is glycolysis. So what is this glycolysis? I told you in ethyl alcohol fermentation also, glycolysis is the process of breaking down one glucose molecule into two pyruvate molecules plus two ATP molecules plus two NADH molecules. As you can see, everything is 2, so it's easy to remember. Now, glucose is a 6-carbon molecule. Pyruvate is a 3-carbon molecule. That is the reason why glucose is being broken down into 2 pyruvate molecules. And another thing that you have to learn is this ATP produced is the net production of ATP. Remember, it is the net production of ATP. What does this mean? It means initially 2 ATP molecules were used up, and finally it gives us 4 ATP molecules. Then if you calculate the net production, it's gonna be 2 ATP molecules, right? Okay, and to do this glycolysis process, we use 2 NAD plus initially. This NAD plus reduces into 2 NADH in the glycolysis process. Right, so that is the first step of lactic acid fermentation. Now, in the second step, glycolysis, the pyruvate produced is directly reduced into lactic acid. Now, in ethyl alcohol fermentation, remember pyruvate was involved in 2 steps. But here, pyruvate is directly reduced into lactic acid. How is it reduced? Actually, NADH plus plays the role here, right? It acts as the reducing agent in this process. This is the reducing agent. And since with the reducing agent a redox reaction takes place, this is going to be our oxidizing agent, right? So pyruvate gets reduced while NADH gets oxidized. So pyruvate gets reduced by taking the H ion over here, right, into lactic acid. And NADH is going to get oxidized to NAD plus. Since we know that NAD plus is limited in the cells, we need to produce this NAD plus. So NADH is going to get oxidized into NAD plus by removing the hydrogen molecule. So who is the final hydrogen acceptor? The final hydrogen acceptor is going to be the pyruvate molecule, right? This is our final hydrogen acceptor. And remember, in this process, a carbon dioxide molecule is not released because the carbon number doesn't change in pyruvate and lactic acid. Okay? Alright, so now we should know who are the organisms that carry out lactic acid fermentation. Certain fungi and bacteria carry out lactic acid fermentation. Who are these bacteria? These bacteria are the ones which are involved in the formation of the yogurt and curds that we eat, right? We call them the lactobacillus species, or in general you can call it lactic acid bacteria, right? Lactobacillus species or lactic acid bacteria. And also we know in athletes, in the muscles, lactic acid is produced, and that is due to lactic acid fermentation when enough oxygen is not there to carry out aerobic respiration. So in muscles, the anaerobic respiration takes place. Lactic acid fermentation is a type of anaerobic respiration. Therefore, lactic acid is produced in the muscles, and if too much lactic acid is going to get produced, the muscles of that athletes would cramp. So hope you understood the lesson today. Please subscribe the channel if you haven't subscribed to look out for more biology videos like this.